Okay, good morning. Let's start. So far what we have spoken about is population genetics, in which we said things are at a genetic level. We worried about this locus, that locus. We didn't really spoke about things like dominance, epistasis, but of course they are there, right? And Deepa told you about DFEs and so on, but everything is at genetic level, okay? But of course, selection acts on the phenotype. So that tiger, which is going to eat one of you, will not look at your, check out your DNA sequence to see if you are a delicious meal, right? It will just see you, your phenotype, right? Okay, so the studies that we spoke about in population genetics, I said, okay, A1 has fitness 1, A2 has a fitness 1 plus S. So I'm directly working in the genotypic space, in the genetic space, right? So, but as I said already, things happen at the phenotypic level and they connect to genotype, right? So today, briefly, I will speak about what happens at the phenotypic level, and the theory which describes that is the quantitative genetics. And the key point is that it does not delve into the details of the genetics. It's an older theory, I mean, like, again, as much as old as population genetics, but people use it and make a lot of progress. So you and I think maybe a couple of others who use quantitative genetics to learn things, it's used in, for example, animal breeding and many other things. So I'll try to give you some flavor of it, and then tomorrow I'll try to connect the genetic level and the phenotypic level. So we'll get there slowly. Okay, so as Shweta had already set the stage for us, so now we know that these traits, we can have two extremes, which are these monogenic traits. Can you give me an example of a monogenic trait? Let's see from this half. Give me an example of monogenic trait. That means not controlled, affected by a single gene. From this course, any of the other courses, or from your own work, give me an example. Huh? Sickle cell anemia, very good. Yeah. What about some trait which is controlled by a large number of genes from this half? Height. Whose height? Specifically? Human height. That's a classic example. Any other example? Eye color, our eye color? Is it? Is it? I don't know. I thought it was sort of oligogenic. Few loci or many? Few. It's like maybe not 1, 4, 5, 6, but not thousands, as far as I know, right? We saw Drosophila, eye color, Burey's experiment, just 1, right? Okay, so therefore there's a huge variety. Now, however, the evidence is that most traits are not monogenic. Mendel's experiment, that's a classical example, right? They're all monogenic, just 1, and very clear phenotypes, right? Okay, but things like human height and many other things. Another one, some of you are Drosophila people. Drosophila people, can you raise your hand? Give me an example from Drosophila. Huh? Polygenic. Which trait is a quantitative trait in Drosophila? Wing size, yeah. And when I started to think about quantitative traits, everybody said, example is bristles on the abdomen of the Drosophila. I mean, you have to put this fly like this and count the bristles. Okay, anyway, so these are all quantitative traits. And now what people think is that many traits of interest are actually polygenic or quantitative. And there is this theory called the quantitative genetics. Okay, so let's learn a little bit about it today. So we have this monogenic traits, and the other extreme is polygenic, and some people also say omnigenic, that everything matters, which I don't fully understand. And in between is sort of oligogenic, okay? So this is one-ish, and this 1,000 or something like that, okay? So I want to describe that, but the key point that let me again emphasize, we will not, final answers that we get will not have genetic details, okay? Okay, so let's start with that. So let's take this population and let's assume for now that let's say all of us are genetically identical. This is an isogenic population for simplicity, I mean for a reason, okay? And then I count, we measure our height. So we have the same genotype, all of us have identical genotype. But do you think our heights are same? No, right? Okay. So this fact that, so why are our heights different? Why do you think so? I mean, although maybe we are genetically identical. Huh? So let me hear from others. Environments, right? So now it looks very obvious, right? That environment matters, nature versus nurture and so on and so forth, right? But at that time it was not known. At that time means late 1800s and early 1900s, and there's a bitter debate about what matters and how it's quantitative traits and so on. So let me not go into that. But this gives us a simple equation, which you will always see in quantitative genetics. So it's called P equals G plus E. So what does this mean? So what I'm thinking of is, let's take an individual, P1, okay? And we have a collection of these individuals, they have the same genome. And I'm thinking of height, let's say. So let's say we have these alleles T, 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 T. So it's an individual. All these individuals have at the first locus T, second locus T, and so on, okay, for simplicity. So then I measure the height of the first individual with this, okay, second individual. Then as we said, it will be different, right? So what this P equal to G plus E is saying is that for that individual, the phenotype in this isogenic population is equal to something called genotypic value, okay, which is simply the average phenotype for that specific genotype, okay? So this is G of T, 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 okay, which is same as average over P1 plus P2 plus P3, Pn. Okay, that's G, that's what G is, plus something E, which is environmental deviation. So what it's saying is, assuming this population is isogenic, we have same genotype, we calculate everybody's height, I measure, calculate the average, that's G. And the deviation of our height from that average is the environmental stuff, that's E. That's all is there in this equation. But this is a fundamental key equation and we'll use it again and again. Yeah. Yeah.
Are you saying that it's the same gene in multiple loci?
No, it need not be. I just took it as, so let's say we have this individuals. So let's say this in this population, in this room, first locus is T, second locus is a short locus, third is T, and so on. I just wrote T, 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 it's just easy to remember. Yeah? Is this funda clear? I mean, if I think about it, it doesn't look like a great funda somehow, I don't know. Because we do know environment matters, right? But I'll show you one example from Galton, which is in late 1800s. And then maybe you can appreciate that better, that why is it so important? So that's all, P equal to G plus E. Okay, everyone? But of course, you can repeat this exercise for all the genotypes, right? Okay, so let's see how does that matter and why is this interesting and important. Okay, so let's say for simplicity to start with, so this is our P equal to G plus E. Now, let me introduce the other notion, which is of a map. Map, don't worry too much, don't think too much math. What I just mean is, how, if I give you some height, some phenotype, how does it connect with the gene? What is in the genetic detail? Now, you might wonder, but she just told me that we will not do any genetics, why is she bringing that? Well, it has to be. Evolution requires heritability. It requires inheritance, right? So it has to be there. But what's going to emerge is, I will put in some genetic stuff, but what will come out finally will not have any details about these assumptions, about these genetic details. So just stay with me, and I hope I can convey this clearly. Okay, so this map, Shweta already introduced to you, and we will use what is called additive map. So I just wanted to elaborate that a bit. What does that mean? So I have, so what does this mean? So I'm thinking that the height of, let's say I have a single locus model, okay? And there is TT, TS, and SS. So this height of this guy is, let's say the effect, the average effect of the T allele at the first locus is say gamma, okay? And additive means second one also is say gamma, it doesn't have to be. So let's say gamma 1 or the first one, it's with a plus sign, and gamma 2 is this. That means if I have a T allele here, it increases the height by an amount gamma 1. Second also affects by gamma 2. Right, say right now simplicity, that's the equal effects. So Shweta told you this yesterday. So but I have TS, this is gamma, and let's say short allele decreases the height by the same amount. It's just an assumption, it's not really important, you can generalize it. And similarly, this. So that's the additive map, plus E stuff, which is very important. Okay, that's additive map. How about this one? When I say that, Shweta was saying yesterday, we neglect dominance epistasis. What does it mean? Let's just get that clear. So suppose I have PTS, and I say there's dominance. What does that mean? Yeah, the effect size, not P, yeah, effect size. Effect of TS is not equal to effect of TT okay? When this is true, then I have dominance. In this example, do you think I have dominance or not? Sorry? Yeah. No, dominance is, how do you find dominance? You look at the average effect of the two homozygotes. So the average effect of two homozygotes is 2 gamma. Average effect of the other homozygote is minus 2 gamma. If I average it, 2 gamma plus minus 2 gamma by 2, that gives me 0. That's what I wrote over here, right? So in this one, it's an additive map which neglects dominance, right? So this is the dominance. If this is not true, then we'll say we have dominance. What about epistasis? For epistasis, I need to introduce at least one more locus. So this was one locus. Let's do one more locus. So say I have TT at the first locus and ST at the second locus. Yeah? This, if I, sorry? When dominance is there, then it's a continuum. If there's no dominance, then the fitness would be intermediate of the two homozygotes. Then there's no dominance, right? Or the semi-dominance, right? So I'm saying when then the average of the two with the same concept, average is not the same as that heterozygote, then there is dominance. Clear? Okay. What about epistasis? Anybody knows what the meaning of epistasis? But we are not asking experts. We want to ask the wannabe experts. Huh? Yeah, interaction, but what? So I want to make it slightly more quantitative in the sense that precisely what does it mean? So this effect is equal to first locus. This is epistasis. This is epistasis. That last line. Yes or no? Yeah? So what is epistasis? Dominance is, what I just described to you at the same locus by the two alleles. Epistasis is the interaction between the loci, right? So that's why I say I need at least 2 loci. So at the first locus we have T1, T1. Second locus we have S2, T2. So if this effect is as though I can treat them independently, effect of this guy plus effect of this guy, then there is or there is not epistasis. No epistasis. So this is not epistasis. right? Yeah, if it's not equal, then there's epistasis. Is this much okay? So I'm going to ignore this because it's a Thursday, also because I don't know. So we will just stick with that simple thing. And I think Shweta will also, right? Good. Okay, so, but it's good to know what assumptions we are making because they do matter. For the human genetics, as Shweta already pointed out, this is a good enough model. But for others, like somebody was saying, plants and all, it may not be. Okay, but it's good to know. Yeah? So keep this at the back of mind and we will stick with that one. Now, I want to know a very simple question which will be based on two aspects, two things I will use. One is our P equal to G plus E and the fact that we have a quantitative or the polygenic trait. What does it mean? It's controlled by a very large number of loci. So let's say number of loci is L, which is very, very large. Lots and lots of them. Okay? So I want to ask this question that, we said that we are genotypically identical, but most likely we are not. And again, I'll take Shweta's example in which, this row has, same genetically identical, these guys, and so on. But let's pull all of our data of our height. And I want to know what's the distribution of our heights. Probability of height in this room, that is the distribution of height. Any guesses? Huh? What? Normal distribution, I don't know. Why everybody likes normal? So what's the normal distribution, by the way? What's the normal distribution? Normal? When you say something in normal distribution? Say something more, huh? Yeah, but okay, shape is like this, no? That bell-shaped curve. So, I live on this something called New Bay Road. So we have a new restaurant opening there. It's called the Bell Curve. Everybody knows. I mean, I was like so I'm impressed. People know about Bell Curve. Okay, that's a sort of fancy restaurant now. Okay, so so you're saying because you sort of intuitively expect this. A couple of comments here. Just because you see something like this, it does not mean it's normal or Gaussian. Okay, there are all kinds of distributions which will look like this. So be precise about what distribution you're talking about. Just because it looks qualitatively like this does not mean it's a normal distribution. It's a very specific function, very specific properties. Okay, but what you're saying is right. You expected normal distribution, and that's something which we should have done in the class. And I think it's one of the projects. Okay, and you will find it's a normal distribution, but can we understand that? So this has been done again for height, for this abdominal bristles and so on. Lots of quantitative traits, they're all normal. Sometimes you have to do some sort of a transformation of variables, but they all become normal. Can we understand that? Is there some basic funda? Okay, so let's see if we can get you the funda. So let's do this exercise. Actually, we have done that exercise, the coin tossing experiment of ours. What happened in that experiment? Let's recall. So this is what you did for your tutorial, right? So each of you had tossed a coin, okay? Let's say L times. Tossed a fair coin. This L is same as that L, okay? L times. And I say that when you get a head, you put a value 0. If you get a tail, 1. And if you get a tail, you put a value 0. And then you counted the number of heads. Can you say that same thing in terms of ones and zeros? What does that mean? So whenever I got a head, I put a 1. Whenever I get a tail, I get a 0. So when you measured the number of heads, what does that mean in terms of these ones and zeros?
The probability of getting how many times out of the
Yeah, okay, so maybe I'm not so clear. Let me explain that. So number of heads is what you were measuring, right? Okay, So if I now think of, so slightly abstract, but it's a very useful thing, and physics, chemistry, biology, mathematics, whatever, it's a very, very important thing, and you're using this actually all the time, especially the experimentalists, or if you do simulations. So I'll first introduce the word and explain it. So it's called central limit theorem. I think it's sort of early 1900s or late 1800s. So let me explain that. So when you're asking for this, okay, so what are you saying? That I got, let's say, capital X number of heads, right? That's the distribution you were doing. You did 40 tosses, 50 tosses, right? Sometimes you got 21 heads, 23 heads, and so on, right? Okay, so think of it as a sum of L random variables. What does it mean? First time, if I get a head, I put a 1. If I get a tail, I put a 0. But what does it mean that little x is a random variable? Because I get head with a probability half, I get tail with a probability half, right? So the first time, whether I write a 1 or 0, that is random. Is it clear? Probably not. But the key thing is what you did in your coin tossing experiment. That's a very useful thing to keep in mind. Okay? So then this theorem says that when you are adding, when you said head, I said count the number of heads. So that means you're putting a 1 there. Every time you have a head, 1. Head, 1. Tail, 0. Another head. So you're actually, what you're doing is counting, you're adding these random variables. And this theorem says, central limit theorem says, that if this little xi, x1, x2, x3, I write a statement for you, are chosen from a nice distribution. Let's not worry about what nice means. Okay, these are the elementary things. This is our coin. Okay, then the distribution of the sum is a Gaussian or normal. This is what you have done. When you did this coin tossing experiment, all of you were saying, no, Gaussian, normal, and so on. This is actually a very powerful theorem. When you're doing experiments, your advisor say, oh, you just did 3 experiments, look at this error bars, they're huge. Where is that funda coming from? Central limit theorem. Okay? Irrespective of what you do, you have to know this. I mean, you cannot get a PhD, MS and all if you don't know this theorem. At least the statement you should understand, forget the proof, but you have to know this. This is, I mean, I'll flunk you if I come for your defense and you don't answer this question. This is really, really important, I mean, irrespective of what you do. Okay, so that's the central limit theorem. And now, this was a heads and tails and so on. What has that to do with our problem? Forgot to say, nice distribution of sum of large number of random variables. Large number. This lots of them we have to add, right? Because you did for 40 of us, no, it looked nothing like the normal distribution, right? Or you did sort of 50, right? Okay. So now, what has that to do with quantitative genetics? We said it's a quantitative trait. Large number of variables. Large number of random variables. So that's because it's a quantitative trait, right? What is this random? I am calculating this P of gamma, maybe it's gamma for TTTTT, right? Plus some E, right? Where is randomness in this? I'm adding this lots of gammas, no? 100 gammas plus E. How is that a random variable? Is it a random variable? Because E is the random variable. So what you can think of it is E is such that if suppose the gamma, the effect was 1, okay, E has this effect that it will give a kick of 0.1 with the probability half, and minus 0.1 with probability half. You're coin tossing. This is the simplest model of E, right? Sometimes you'll get 0.9. Sometimes means what? With the probability half. Sometimes you'll get 1.1 with the probability half. So what you're doing is essentially what I wrote over there. Exactly what you did for a coin tossing problem. So there you're getting 1 or 0. Here is 0.9 and 1.1. So sometimes means some individual. Right? And lots of them. So you're adding this large number of random variables, and therefore central limit theorem applies, and therefore the distribution of height is a Gaussian. Distribution of abdominal bristles is a Gaussian. That's the sort of very important thing here. So what did we do therefore? We use this little two ingredients, P equals to G plus E, adding up, E being a very crucial component, and the fact it's a quantitative trait, and therefore we're adding a large number of random variables. So digest it for 1 minute. Again, feel free to chat. Okay? Yeah.
Use this to argue that about distributions that are not Gaussian. So if they are derived from non-random variables.
No, but then if they're not random, but then why would they be distribution? All of us have no effect of environment. We have genetically identical, we also have same environment. There's no effect of environment, let's say. We have the same answer, we all are 5 feet tall. What is the distribution, right? So randomness is essential. Yeah, right? So his question is, what about non-Gaussian distributions? Of what? Of the sum? By adding these things, can we get non-Gaussian distributions? It's possible, but I think it's probably not pertinent to this discussion. But in physics, we do encounter, and they are very interesting, for us at least. Okay? Anyway, okay. And just a small corollary from this is, and which I'll need, is the following, that if we now look at the variance, everybody understand what is variance? Okay. So one corollary of this P equal to G plus E, which I'll just write, and you should think about it, assuming things are independent. So I have removed dominance, epistasis, and I'm also assuming that some G cross E interactions are absent. Then the variance in the phenotype is equal to variance in this, or rather the additive variance in the genotype plus variance of the environment. So this follows from P equal to G plus E under independent, things are independent type of assumption. Okay, so just try that. I'll need it, so please box it and keep it. Okay, so now, as I said in the beginning, P equal to G plus E looks so obvious. There is an effective environment. So what's so important about it? So I'll give you this example. Okay, so this is 1886, I think. So I think Galton, everybody has heard of Galton? Anybody who has heard of Galton? Francis Galton, yeah, that's his first name. What else have you heard about him besides his first name? Huh? Huh? Yes, sort of foundational, yeah. He was also some cousin or something of Darwin, right? And there is a lot of math also he did. and his regression that Shweta pointed out, he devised those methods. So you can blame him if you find it difficult. Okay? So this is like a little experiment. And again, I think this was being asked as a project. And let me ask you what you think. Suppose each of us find, our height and mid-parent height. What does that mean? So he goes to home and asks his dad and mom, find their heights and take the average of the height, and finds they have some certain height, and corresponding to their height, he has some height. Then he does the same, he does the same, she does the same, she does the same, and so on. All of us do that, yeah. It's the average height of the both parents, yeah. So dad and mom's height, averaged. Okay, yeah, okay. So some of us would have the same mid-parent height, average height of our parents is same, but of course, my height, maybe my parents' height, average height is same as his height, but clearly he's much taller than me, right? So that's you, right? Okay, like that. So for each corresponding mid-parent height, you expect there will be some variation, right? Okay. So let me try to draw this. So we expect something like this. Some points here, and I'm not drawing it nicely. I should have tell you how many such points are there, right? Okay, so what do you expect on an average? Okay, let's see here. On an average, you expect that higher the mid-parent height, more points over here, right? Because you know genetics, you know about heredity, right? Typically, that's what you expect. Of course, if my parents are, it's possible I'm shorter than both my parents. It's possible, right? So expect this kind of correlation, right? So there is some scatter along this, right? But suppose I take all these average over all these heights. Where do you think that average will fall? Will it fall on this line, which is exactly exactly diagonal of this square? Is the question clear? So let's say the mid-parent height is 150 centimeter. Some have, so this point is also 150. That's this diagonal line, right? So some have height 170, some have maybe 135, and so on. If I average all these ones, do you think that average would be 150? What's your expectation? No. Right now, I don't know, I'm in 1886. Forget P equal to G+E. Go just after a little bit Darwin. The work that I'm describing to you is in 1918. So this is 30, 40 years before Ronald Fisher, who laid the foundations of how these two things, Mendelian genetics, is compatible with quantitative genetics. Okay, so let's forget P equal to G+E, we don't know. But what's your naive expectation? What do you think? If average height, if I take the set of people in this room whose mid-parent height is 150 centimeter, and here there's some scatter, and we average over heights. So his parents' height is, average height is 150, mine is also 150, and his is, and hers also. And then we average our 4 heights. Do you think it will be 150? Naively, I would think yes. But that's because we are naive, at least at that time, right? That's what we would expect. But when he actually did the experiment means collecting the data, he found that the best fit, and I'll say a little bit about that, the slope of that best fit was below the diagonal. Diagonal has a slope 1, right? So this slope is less than 1. And he colorfully described it as a regression towards mediocrity. That is, somehow, I don't know, the slope is smaller. That means the population is regressing, not progressing, opposite of that, towards the mean population, that is mediocrity. Okay? So this is called regression towards mean more generally. Okay? So we want to understand, this is 1886, before I already showed you the 1918 stuff. And he doesn't know why. He expected, I think from what I remember, he expected a diagonal line. So now it's obvious to us it should be P plus E equal to G plus E. But it's not so clear that the slope should be less than 1. And now you sort of see that why it would be less than 1. Do you see it? I don't know, I'm not sure if I see it. Yeah, it need not be exactly same, but yeah, it could be different, but I don't know why it should be below. Okay, so let's do a little bit more work. Okay, so since we're running out of time, I will merely give you the answer. So I'll just merely sketch it for you. So key is P equal to G plus E. Using this for the additive model, you will get this VP equal to VA plus VE, as I said already. You have to show it, you have to sit down and do it. But if I want to explain this kind of data, which is between two generations, okay? It's not about a variance in a generation, it's between two different generations. Then what you need to calculate is something called covariance. Everybody understand what's the covariance? People know what's the variance, right? If I have some variable x, random variable x, so I'm not sure what notation you guys use. Do you know what it is? This minus summation over x1 square, x2 square, and so on, minus average squared, right? That's the variance. Covariance between two x and y is something like this, but xi yi minus subtract the average of these two. That's called a covariance. Covary, two guys, two random variables, okay? So you need to calculate the covariance between the parent and the offspring. We will not do that. And what comes out, you will sit down and do it. It's not a difficult calculation, but you still have to sit and work out. So covariance of the parent, mid-parent, and the offspring, it turns out to be VA by 2. VA is that genetic variance, the additive component of the genetic variance, okay? Okay, so there is this. One more bit, just bear with me. Again, I think Shweta already introduced, but I will just very quickly summarize. She said this best fit, which is a line of regression. What does that mean? It means that if I have y equal to alpha, I forget what was her notation, I think, plus beta 1x, something like that, beta and alpha maybe, alpha plus beta? That's correct, okay. So this is the actual data, y, these points. And alpha plus beta naught plus beta 1x is that line. Of course, it's not same, no? So what we would like to do is to minimize the error between the two. So that's the regression method. So if you do that, then it turns out that beta 1 is nothing but covariance of x and y divided by variance of x. So this is all I'm just giving you. It's an overview because we are running out of time and you're tired. Okay? And if you use this and variance of x, which is the variance of the phenotype of the parent, so this one is fitting. So I'm trying to understand the meaning of this line. It's all nice, math, statistics, that's cool. If you work out all this, it turns out that this slope has a biological meaning. It is equal to additive variance divided by VA plus VE. That's biological meaning of that slope. And this is called heritability. More precisely, narrow sense heritability. So what we are calling slope, that has this meaning, biological meaning, that it says that this variation, what component of that, what fraction of that is explained by genetics, specifically additive genetics under the additive model. Okay? So heritability of some traits is small, some traits is large. For example, for human height, it's pretty large, 0.7, 0.8. Okay? Can you tell me, okay, that line, the dashed line, what's the heritability of that line? Huh? No, dashed line. It will be 1. If there was no effective environment, VE is 0. So then only, right? So when VE is 0, there is no effective environment, it is perfectly correlated, the parent and the offspring. Then only you will get, but of course that's not true. And no wonder slope is less than 1. Okay? So that's sort of roughly the idea. Okay, so fine, let's go ahead anyway. Yeah. Yeah, let's see. And this, fine, we got some numbers, some data, and so on. So what? One more comment I'll make in this case. Suppose you're a cattle breeder, okay, and because this thing is used a lot in animal breeding and plant breeding, right? Suppose you're a cattle breeder, you have the milk production has it's a quantitative trait, so it's a normal distribution. But you don't want this less milk-giving cattle for the next population, right, for the next generation. What would you do? You would say that, okay, this is a distribution. X-axis is the milk production, and this is distribution. As we said, it's normal. You will maybe pick out top 1%, 5% to find the next generation. That's called truncation selection. So the original population had certain mean, and now I'm taking the top 5%. So the mean of the new population, which is going to find the next population, has changed. Okay? So that's called S, or the selection differential. The question is, what happens now? Suppose I had not done this truncation selection, then the offspring will also be same, no? They will also be normally distributed. But now I'm doing this selection. What is the effect of that on the offspring generation? What will they be like? What will the milk production be like? No, you expect the milk production will be higher, right? Because now you are choosing the top guys to find this, right? So there's a little equation, R equal to H square times S, which says this is the selection, which you have used to find these new guys, and this is H square which I just defined, and that will tell you that the new guys, the offspring, the next cattle, how much milk production would increase by what amount. So now you see how useful it is, and there's no gamma in all this stuff. R equal to H square S. So how will you do? I don't know. I'm a farmhand. I don't know anything about gamma and additive and this and that. How exactly would you use this? So what I will do is, I will do this kind of stuff. I have a lot of population. I'll do the mid-parent height. I'll do the offspring height. Find H square by this regression. I got H square. I know how I've done the truncation. And I know what to expect. Should I pick 5%? Should I pick 2%? Should I pick 10%? Okay, so that's the real funda there. And it is very useful, very helpful. Okay? That's the idea. So when I said it's a macroscopic theory which does not care about genetic details, I wrote all the gamma, shamma, and so on. But not there, no? Okay? So that's, and everything is about its variances. That's the key quantity. And if you're a physicist, it's like thermodynamics. You worry about things like pressure and so on. These are macroscopic variables. You don't know what these microstates are. Okay? Fine. So we can do all that. How many are happy with this? This equation, by the way, is useful for short-term selection. It does not work if you want to continue doing this for ages and ages. Okay, so but let's say we can do all that. How many of you are happy with this? Yeah. S is the selection differential, which is the mean of the population, and after truncation selection, when I picked up 5%, the mean of the new population. So that changes the selection. That changes H, and H square I got from there. How many are happy with this? All this is great, wonderful. But is there anybody, something nagging you, something, your fingers are itching to do something? Anybody? Happy, everybody? Oh wow. Huh? Still processing, okay. So 2 minutes of processing. No, I want you to understand what I want to say next. That why do it? Why care? So therefore, 2 minutes, it's 11:34. Please process while I clean this. Oh. So why not stop here and sort of massage the theory and go home? Is there anything more? Can we move beyond 1918? Is there a reason? How do we push those boundaries, those research questions? Is something, or everything is good, all done, everything I know? Is it? You have to think. No, there are lots of reasons. Lots of, because it's quantitative by definition. I'm saying about this not specific equation or something, but this general setup that I said in which you worry about this variance, which is a macroscopic quantity. You use it and so on. Yeah, that we can do. Yeah, sure. I mean, I don't think V is known. I mean, I'm not sure how that's done, but generally what we know is this ratio. That's as far as I know. Right? But is this still something bugging you? No. Hey guys, come on. I taught you 2 classes before. Something should be bothering you. Should be moving around in your chairs. Okay. Somebody bothered? Oh wow, who's that? Santana. No, he better bother because he's in my lab. Anybody else? Yeah.
The left side was the heights of the participants and the right side was the height of the...
Right side means x-axis? No.
No, there were 2 distributions.
Oh.
On the right side.
No, there was no distribution. No. Yeah, I'll explain it again later. No, that's not distribution. I'll tell you later again. Yeah. Anybody? What is the VA? How do I find the VA? I can do it phenomenologically. But how does it depend on mutations, selection, DFE, mutational bias, drift? Is it not bothering you? What's going on at this genetic level? That's the sort of the microscopic stuff, right? Right? I can tell you the room temperature here, fine, and there are lots and lots of molecules. But you still probably want to know, maybe not, how these molecules are behaving. Why is the temperature T and not something else? What's maintaining it? What's going on? I don't know. How are these allele frequencies changing? Are they changing? More generally, what kind of genetic architecture that quantitative genetics is assuming? It works, yes. But what's this underlying funda? What's the underlying genetic stuff? We said dominance epistasis, but still, what's going on there? What's the genetic architecture it's assuming? So that it's valid in those conditions. And if those conditions are not valid, quantitative genetics will not work, right? So we need to know when does it work and what is it that we have implicitly assumed. So this, again, Fisher in 1918 had thought about, so I'll explain that. And then we'll be done. Okay. So again, we stick with our additive model, and I say that suppose because we started population genetics, we'll go back and look at, let's say, one locus, and so it's a diploid. So we have this TT, TS, SS, right? let's say the probability, the frequency of T is P and Q is the frequency of allele S, okay? So then the frequency of TT is, we did not discuss unfortunately, P square, but it assumes something called Hardy-Weinberg equilibrium. We did not get a chance to discuss that. So under Hardy-Weinberg equilibrium, this frequency of the genotype is a product of the frequencies of each allele. So this is 2PQ because ST or TS. Okay. So then, if I want to find the mean trait, and I'll tell you why I'm writing all this. How will I do? I have an additive model. Okay. So what's the mean phenotype? The frequency of this type is P square. And what's the effect? 2 gamma. That's how we calculate mean, no? Frequency times the value, right? Summation Pixi plus Q square minus 2 gamma plus 2PQ 0, right? Because we said gamma minus gamma, right? So that will give me plus Q2 P minus Q times gamma. Okay, it's a very silly calculation, but it's useful, so that's why I'm doing this. Okay, so this is mean phenotype. Let's just keep it. And because everything is additive, independent, and so on, for L loci, Z is 2L gamma P minus Q. I'll not bother you too much, but you can do this calculation again and find the additive variance, yeah? P square minus Q square, but P square minus Q square is going to P plus Q into P minus Q, and P plus Q is 1, so therefore it's P minus Q. Okay, and using this, you can find additive variance, which comes out to be L gamma square into PQ. Okay, this is elementary math. Okay, you have to do it. Okay, now, coming back to this question. When is quantitative genetics work? What kind of architecture, genetic architecture is implicitly assuming? Okay, so here are the two considerations. First is that, okay, we are doing some adaptation, okay, population is adapting, variance, there is some variance and there is some Z, that's all we bother about, right, in the quantitative genetics picture. So if I assume that, let's say which one comes first, yeah, so as a result of these two, I'll just write the answer for you instead of bothering you too much. So using these two, it turns out okay, Z is the mean phenotype, L is the number of loci, yeah, okay. So here are the considerations. So please just let me, do this with you. Okay. So suppose I assume that, variance does not change. Increase with L. See, variance is there, no? At each locus there is some T loci, some S, some T. Now I have 100 loci. There is some variation because it increases with L. 100 times some number. If I have 1,000 times that number. So as I increase number of loci, because I'm going to a quantitative trait, which has lots and lots of loci, variance will keep increasing. Does that make sense? Variance should increase with L. It should increase with L, yes, but it will be just increasing away. It will become infinite if L is huge, right? So one consideration is that variance is some number, something, I don't know, 2, 3, 4, something like that, not a million, okay? In other words, it's a finite number, okay? If it does not increase with L, then VA is a constant with L. What that means is, this is slightly more than what you might know. What that means is that gamma should decrease. So as L increases, VA increases. But if gamma decreases, the effect size decreases, then my VA will not change with L. I'll write it for you. You can think about it again later. So gamma should decrease as with L to keep VA independent of L. Okay, so what does that mean? This is all that stuff about small effects. L is very large, 1,000, million, and so on. That means the effect of each of them in the quantitative genetics picture is that effect is independent, effects are small. So this quantitative trait at height is being affected by a large number of loci, each with a tiny effect. If you understand this equation, that's what it means. If I increase L, effect size decreases. So this is where this small effects we keep saying, that's where it comes from. Okay? So this coming from this consideration that the additive variance is a constant. It also leads to the following thing. It also means, second consideration if I have is, that change in phenotype, it also cannot be huge something. It's something finite again. It cannot be Hajas standard deviations. It's something finite. If I do that, second consideration is phenotypic mean deviation is also finite. If I use that, then using this, it follows that the change in allele frequency also decreases. So what one is saying here is that allele frequency is changing, but there's subtle changes in allele frequency. We are not going to look, because quantitative genetics guys, they are not looking at the allele frequencies. But implicitly one is assuming that the quantitative trait is is affected by a very large number of loci, and the allele frequencies of T and S for a very large L is only subtle changes. Okay, so there are teeny changes, but lots and lots of them, such that still the milk production will increase, such that you don't lose the variance. Okay, so that's the genetic architecture which is being implicitly assumed here. But as time goes on, because L frequency is changing, tiny, 10 to the minus 6, but if you wait long enough, those tiny changes will accumulate, right? And these assumptions that the variance does not change with time, it does not increase with L and so on, they break down. And that's why you cannot use quantitative genetics for long-term prediction. They are valid and reasonably good when you are doing short-term predictions, few generations. Beyond that, you cannot do. So, I want to understand Lansky experiment. That's a long-term experiment. I cannot use quantitative genetics, right? So therefore, we have to go back and look at the full genetics, full population genetics and quantitative genetics, and see if you can build models or have some understanding of these two together. There are several models, and I will explain one to you tomorrow. And yeah, so Shweta is going to now talk next. And suddenly, why are we so much interested in all this stuff? We didn't care in the early 1900s, right? Suddenly we care because now we know. Now we know allele frequencies, how they behave. Now we know what are these small effects. Are they really small? Are all of them small? What fraction is small? Can they be large? How large is large? How do they affect adaptation? These large effect loci, which she'll explain, do they affect anything in our dynamics? Okay, so that information, because genetic sequences, which is just 15 years or so, and GWAS, that's giving us all that information. So there's a really hot area in which people now want to use this new information coupled with what we know from quantitative genetics and a lot of population genetics to bring this rather complex models, but hopefully towards that prediction, okay? Which at least to me, as a physicist, is very satisfactory because you're integrating this microscopic and macroscopic. Okay, with that I will stop. Thank you very much. Any questions? Yeah. Oh, okay. So frequency of TT into its effect plus frequency of this, the way we do any calculation of mean, probability distribution times the random variable. So that probability distribution is this frequency and effect. Frequency and effect, frequency and effect. That's what I was asking. P plus Q is 1. Maybe I didn't say that. P plus Q, because either you have S or T. P plus Q is 1. Sorry, I forgot to say that. Yeah? Okay. So therefore, tomorrow, I will put some of these things together and explain to you some of our work, which combines these things. Okay? Thank you.